This is the second in the series of 3 videos looking at how photosynthesis works in plants, and we're now going to start delving into the detail of photosynthesis by looking at what occurs in the first stage, the light-dependent reactions. First question to consider is where do the light-dependent reactions happen in the chloroplast? The answer is it's the thylakoids that are responsible for these reactions. Those were the discs that you saw in stacks in the cut open diagram of the chloroplast. And you can see a cross-section of a thylakoid disc shown in the diagram, rather complicated looking diagram here. And this represents the fact that the thylakoid itself has a membrane shown as the kind of grey and orange band going around the outside that encloses a space within filled with an aqueous solution called the thylakoid lumen. You can see that embedded within the membrane of the thylakoid are a whole collection of fairly complicated looking structures, and it's these that are responsible for carrying out the sequence of steps needed to produce the ATP and NADPH that are the necessary products from the light-dependent reactions. If you're looking at the diagram thinking, wow, that doesn't make any sense at all, the rest of this video will unpack the individual steps, and hopefully it'll all come clear. So we'll work our way through the sequence of steps as a kind of a story, and the story starts then, rather strangely, with something called Photosystem II. There's a Photosystem I as well, which was discovered first, and hence how it got its name. But the story starts at Photosystem II, circled in the diagram here, and we're going to sort of zoom in and consider what happens there in some detail. So our story starts with a packet of light energy known as a photon, probably from the sun, entering the leaf and hitting photosystem II. The light energy will be absorbed by one of the pigment molecules, and these pigment molecules can pass on that energy until it reaches that special chlorophyll molecule in the middle, the P680. The effect of the light energy when it reaches the P680 is to excite an electron. Exciting an electron simply means giving an electron energy. So the energy ends up being collected by an electron. And this process of giving energy from light to an electron has a special name. It's called photoexcitation. Photo here not meaning anything to do with cameras, just meaning light causing excitation. So the light energy has been captured by pigment molecules, passed to the P680, where it is gained by an electron. That electron then actually leaves the P680 and is passed, you can see this represented by the orange arrow going up in the middle, onto a structure, to part of the photosystem II called the primary acceptor, which then transfers it onto something called a mobile electron carrier protein, specifically called plastoquinone, in this case labelled PQ in the diagram. This is literally a protein that is able to transport that electron on away from photosystem II to the next step in the story, where it's going to make a process happen. P680 is changed by this. Actually, it loses 2 electrons this way, and if you lose electrons, you become a positive ion. So the result of having lost electrons is that the P680 is now a positive ion. So this diagram gives a simplified representation of photosystem II, the start of our light-dependent reaction story. In reality, it's made of many specialised molecules, not shown here, but this gives the detail that we need. I want to draw your attention to those green discs. Those are pigment molecules that can absorb light energy, things like chlorophylls and carotenoids. In reality, there's more than one type of chlorophyll. We don't need to think about the detail of that, except to note that at the centre there's a special type of chlorophyll called P680 that's going to play a key role in this first step. Before we look at where this excited electron goes with the mobile electron carrier protein and what it does, we're just going to stop and consider how the P680 plus gets its electrons back again. This is vital because if it couldn't get its electrons back, then it would never again be able to play its essential part at the start of the LDR. And this involves water. And it's at this point that the oxygen that's so important to us as humans, as a waste product of photosynthesis, gets produced. So let's look at the detail of what happens there. The way the P680 plus gets its electrons back is by grabbing them from a water molecule. P680 plus is what's known as an oxidizing agent. Oxidizing agent is something that can take electrons from other things. And in fact, the P680 plus is the strongest oxidizing agent that you meet anywhere in the biochemistry of life. So the P680 plus grabs a couple of electrons from a nearby water molecule to replace its lost electrons. And the result of this is that the water molecule splits up. It splits up into hydrogen ions and H+, also known as protons, and also oxygen, O2. The splitting up of water using a process that started with light has a special name. It's called photolysis. Again, the word photo referring to light, lysis meaning cutting in a way. So this is the cutting up of water using energy that came originally from light. So let's turn our attention back now to our excited electron and follow it on its journey. Remember, it's gained energy from light, and it's been passed on to this carrier protein, the plastoquinone, and this now transports it to the next structure in the thylakoid membrane called the cytochrome complex, where the energy that electron has is now used to carry out a specific process. So let's look at the detail of that next. So the cytochrome complex is another structure embedded in the thylakoid membrane. You can see it stretches from the aqueous solution on the outside of the thylakoid, the stroma, through to the solution on the inside, the thylakoid lumen. Its role in the process is to bring hydrogen ions, H+, from the outside of the thylakoid, from the stroma, through into the inside to the thylakoid lumen. It's described as a proton pump. H+ ions can be called protons, and so it pumps protons from the outside of the thylakoid to the inside. And to do this, it needs energy, and that's where our excited electron comes in. As the excited electron is delivered to the cytochrome complex by the plastoquinone, the carrier protein, it travels through, and its electron is its energy, the energy of that electron is used to operate this proton pump. So it's the energy supplied by our excited electron, the energy it gained from light energy, is now used by the cytochrome complex to pump protons from the outside of the thylakoid, from the stroma, to the inside of the thylakoid lumen. So our electron is losing its energy here. It's no longer excited, and it's picked up on the other side of the cytochrome complex by another one of these mobile electron carrier proteins, this time called plastocyanin, which is going to carry it on to the next stage of our story. Before we look at what happens next to our electrons, though, we're just going to turn our attention to the purpose of these hydrogen ions that have been brought into the thylakoid, and that's going to turn our attention to another structure in the thylakoid membrane called the ATP synthase. So let's look at the detail of what's going on there. So we've had hydrogen ions produced in the thylakoid lumen from the photolysis of water. We've had more hydrogen ions brought into the thylakoid lumen through the cytochrome complex using our excited electron. The result of this is that we've developed a high concentration of hydrogen ions in the thylakoid lumen. This high concentration on the inside of the thylakoid now means that hydrogen ions will be pushed out through another structure embedded in the thylakoid membrane called ATP synthase. And as they travel through ATP synthase, they allow a very important process, or they make a very important process happen. So the purpose of building up this high concentration of hydrogen ions, of using the energy of the electron to bring the hydrogen ions into the thylakoid lumen, is to operate the ATP synthase. The role of ATP synthase is to take ADP, adenosine diphosphate, and to join another phosphate group onto it to make adenosine triphosphate. This is an energy storage molecule, and it's going to be very important in the second stage. The process where hydrogen ions flow out from this high concentration area inside the thylakoid to the stroma outside where there's a low concentration gets the fancy name of chemiosmosis. So the process of chemiosmosis, the hydrogen ions flowing out from the high concentration in the thylakoid to the low concentration in the stroma, drives then the reaction of ADP with a phosphate group to make ATP. And that reaction has its own fancy name. The gaining of a phosphate group onto ADP is called phosphorylation. And because in this set of reactions it's the energy for it originally came from light, we're going to use that word photo again. So in this case, sticking a phosphate group onto ADP is called photophosphorylation of ADP. So we have chemiosmosis driving photophosphorylation. So we'll turn our attention now back to our electron travelling through this electron transport chain. Remember, it's passed through the cytochrome complex. It's been picked up by another mobile carrier protein called plastocyanin. And the next stage on its journey is photosystem 1. So let's look at what happens to our electron when it reaches photosystem 1. So here's a simplified representation of photosystem 1, and it should look pretty familiar to you. It looks rather like photosystem 2, and indeed they are quite similar structures in many ways. They contain many of these pigment molecules, the chlorophylls and the carotenoids. There is one difference. If you look at the centre, you see that rather than the P680 chlorophyll that was in the photosystem 2, photosystem 1 has a P700. But the principle is very similar. Our electron is delivered to photosystem 1 by its carrier protein, the plastocyanin, and is delivered to the P700. Photons of light, these packets of light energy, reaching the leaf can hit the pigment molecules in photosystem 1, and that light energy is delivered to P700, where it's gained by our electrons. So at this point, having lost their energy in the cytochrome complex, running the proton pump there, the electrons are now re-excited. So this is a second photoexcitation stage. The electrons get a fresh boost of energy, and once again they're passed on out of the chlorophyll in the centre, the P700 in this case, to the primary acceptor and delivered on to another mobile carrier protein, this time called ferredoxin. So it's going to take them on for the now final stage in their journey. So what is this final stage of the story for our electrons? It involves another structure embedded in the thylakoid membrane, NADP plus reductase, and let's see the detail of what happens there. So remember, our electron has been excited again in photosystem 1, gained a bunch more energy, and is now being delivered by its carrier protein, ferredoxin, from photosystem 1 to this final step in the light-dependent reactions, the NADP plus reductase site in the thylakoid membrane. What does it do there? Well, it helps make a vital process happen, the conversion of NADP plus to NADPH. You don't need to worry about what those letters stand for. It's actually the initials of the name of the substance, nicotinamide adenosine dinucleotide phosphate. I don't think it helps really to use the words instead of the letters. It just represents a particular molecule. So the electrons are essential here. In order to convert NADP plus to NADPH, you need a hydrogen ion, but you also need 2 electrons. So and energy is required as well. So our excited electrons here allow this reaction to happen, converting NADP plus into NADPH. And the NADPH, we will see in the second stage of photosynthesis, has a vital role to play. So at the end of all those quite involved steps, we can actually quite simply say that what's happened as a result of the LDR is that the molecules ATP and NADPH have been produced and sent on out into the stroma, where they're going to play a vital role in the second stage, the light-independent reactions. Also during the LDR, And of no significance to photosynthesis, oxygen has been produced. As far as photosynthesis is concerned, it's a waste product. But of course, for those of us who like to breathe, it's of vital importance. From a chemical point of view, we can summarize the light-dependent reactions with the equations shown here. So we see that overall, 2 water molecules are used to produce an oxygen molecule and some hydrogen ions. We've got our 2 NADP pluses turning into 2 NADPHs, and we've got 3 ADPs, each gaining a phosphate group, shown as a PI, their inorganic phosphate, to make the 3 ATPs.